Thank you, Lars, for the introduction and the invitation to speak to this group. I'm happy to share some of our results and interests in how the cell wall behaves and its nanostructural basis for mechanical properties. And so I title or subtitle this "Insights from a Coarse-Grained Modeling of the Cell Wall," the primary cell wall, and this gives insights into the basis for elasticity and plasticity and yield thresholds. And it's also the background interest is in mechanisms of cell wall loosening and growth, although I won't be talking about that specifically in this seminar, this webinar. The image that you see at the right is something that I published some years ago based upon an AFM scan, and then it was decorated with some coloring pencils to put in some of the features of what we thought at that point for the matrix components. So the cellulose microfibrils are shown in blue, and they're robustly show up in that image, and it's a little bit harder to detect the positions and the conformations of some of the matrix components, although that's of interest in understanding the mechanics of the cell wall. So let me remind you of the motivation. And so it's, for me at least, it's for plant growth. And so I'll remind you that plants emerge from these tiny meristems at the shoot tips and root tips. And so the cells are tiny, and then they elaborate through a period of cell enlargement to make these kinds of structures. This is not quite a tree, but it is as large as maybe some small trees. And this process then involves a period of cell enlargement where the volume increases and the cell surface increases. And so since I'm speaking to a wide audience, I just want to remind you that plant cells synthesize their own cell walls, which are glued together. That means there's no cell migration, so growth and morphogenesis depend upon cell enlargement. And the cell wall is the major physical limitation for plant cell enlargement. And so we're talking about the surface expansion, the aerial expansion of this sort of a structure. This is an AFM image of the onion cell wall, which I'll introduce to you in a couple of minutes. So also a reminder for this wide audience that the parts list of the primary cell walls, it of course contains cellulose, and I assume this group knows a lot about cellulose. It's made, we believe, as maybe 3 nanometer so-called microfibrils. They should be called nanofibrils, but they're much longer, and they have the ability to aggregate laterally into bundles. There's also hemicelluloses, which have the property of binding to cellulose microfibrils, and pectins, which are acidic polysaccharides and gel forming. Some of them are fairly complex, as you see here. The question that I want to raise is, how do these polymers interact to make a strong and extensible cell wall in growing cells? And for this, there have been a number of ideas proposed over the years, and there's still not a consensus in the field. And so I show 3 images that I think represent the major classes of cell wall architecture. So on the left, you see the concept, or I'm depicting the concept of a pectic hydrogel reinforced by cellulose microfibrils. That's an old idea, and it's been revived in recent years by people interested in trying to connect AFM measurements of cell wall mechanics with growth and modeling of growth. Second class of ideas is one in which the matrix is a giant supermolecule. So it's one covalently linked molecule that then binds to the surface of cellulose through xyloglucans. That's the old Albersheim model, which was discarded for a while, but aspects of it have reemerged, and there are still viable points to this model. And then a third idea, the one that's been dominant for at least a couple of decades, is maybe 3 decades, is what I call the tethered network hypothesis, in which cellulose microfibrils are shown as well separated, as are all these other models. That's an assumption that's been implicit in most of the model building of cell walls in this field, in the primary cell wall field. And they're shown linked together by xyloglucan tethers, which presumably are load-bearing tethers. So question is, how do we test these concepts? And I want to show you some of our attempts to test these using mechanics and enzymes and modeling. And so the system that I'll be focusing on today is the onion epidermal cell wall. And I'll remind you also that the outer epidermal wall of growing tissues limits organ growth. So it's an important wall. And in some ways, it has some of the character of some secondary cell walls. So it's a thickened cell wall and it's a polylamellate cell wall. But chemically, of course, it has some distinctions from classical woody type cell walls. So we found we could peel the onion cell wall or the onion epidermis in such a way that the epidermal cells split in the middle, and so we get these strips of single outer epidermal cell walls. The strips can be a centimeter or more or larger, and they're only one cell wall thick. So about, in this case, a highly hydrated 6 micrometer thick cell wall. We can take these wall samples and then put them under an AFM and image, and also do nano indentation experiments. And in parallel, we can clamp these wall strips into extensometers, stretching devices, and measure stress strain mechanical properties and things like creep under a constant load. So it's a versatile system. And so the experiments, before we get to the modeling, the foundation is empirical results. So in these experiments, we combine enzyme treatments with mechanical assays to try to probe cell wall structure. And here we're asking us the following sorts of questions. So which polymers influence cell wall mechanics? Do mechanical assays give equivalent results? So for example, if you see a change in the indentation modulus, does that necessarily correspond to a change in the tensile modulus? Also, how do elastic properties relate to plastic properties and relate to the creep properties of the cell walls? And so for this study, and the citation is shown at the bottom there, we use a series of assays shown on the right here. So the first assay is what we call chemorheological creep, where we take these wall strips and we hold them under constant force. So it's a tensile force. And then we add a protein to the sample, and we look for an increase in the length of the sample. So and we call that a creep. So that's a classic assay that we use to discover these wall loosening proteins that we named expansins. And for expansins, it's a really robust assay. The second assay is one in which we stretch the cell wall, so we measure a stress strain curve. Typically, we do two cycles, and from that we can get an elastic and plastic compliances, which in our definition of the terms is the reciprocal of the modulus. And a third assay is one in which we use the AFM indenter. So a different mode of stretching the cell wall or deforming the cell wall. We use the AFM indenter to stretch, deform the surface, and we get a force indentation curve. And from these curves, we can measure a modulus and look at the difference before and after enzyme treatment to get a type of indentation modulus. And then the fourth assay here is one in which we simply repeatedly scan the cell wall with the AFM, and as we do so, we see between each scan there's some motion of the microfibrils, and we use that lateral motion of the microfibrils to get an assessment of the mobility of the microfibrils, and we develop that into a quantitative assay in this paper. All right, so there are a lot more results in the paper. I'm just going to focus on a small subset of these that I think are particularly useful. And so at the top column here, you see the different treatments, and then going down the rows, we see the different kinds of mechanical assays also directed by these arrows. And so just to go through these relatively quickly without belaboring the point, when we treat with a xyloglucanase, we don't see any changes in these mechanical assays. So xyloglucanase doesn't cause cell walls to creep. It doesn't cause changes in the tensile plastic or elastic compliances. We did not see changes in the nano indentation compliance, and we did not see changes in the microfibril movements. So these results are very similar to the kinds of things we saw some years ago. This is more extensive, but some years ago we found that xyloglucanase treatment did not cause cucumber hypocotyls to creep, but there was another set of bifunctional enzymes that did cause the enzymes to creep, and that was the basis for a notion that we put forward at that point, is that there are certain limiting junctions, we call them biomechanical hotspots, that involve a mixture, an intertwining of sialoglucan and disorganized cellulose that hold things together. I won't talk about that at this. I'm focusing on other aspects. So no effect of xyloglucan digestion. Pectinase did have some effects. So we saw the microfibrils danced around a lot more. There was a big effect on the nano-indentation compliance, but in the tensile assays, so that is creep or plastic and elastic compliances, we did not see any effect. Third treatment here is drysylase. So drysylase is a complex fungal enzyme mixture that digests, practically everything in the cell wall. Of course, the matrix gets digested first, and then eventually cellulose goes. And so here we saw, again, as with the pectate lyase, or excuse me, with the pectate lyase, we saw large changes in the microfibril motions and in the nano-indentation compliance. We also saw some changes in the tensile compliances, but we did not see creep. So even though we're digesting most of the matrix pretty aggressively, we're not seeing the cell walls creep. In contrast, if we switch, if we allow the endogenous alpha expansins to become active by putting the wall in a acidic buffer, so let's say pH 4.5, then we see the wall immediately starts to creep. That's a robust assay, and yet we don't see any changes in the compliances. Only a very small change. This plus sign means a 20% increase. Only a very small change in the nano-indentation compliance, and then we do see changes in microfibril motions. All right, so those are the empirical results. I interpreted these results, these empirical results, as being inconsistent with these common models. So, for example, the xyloglucanase treatment, it seemed to us was inconsistent with this tethered network hypothesis. The pectate lyase treatments seemed to be inconsistent with the idea of the wall being a hydrogel, simply a pectic hydrogel reinforced by cellulose microfibrils, and so on. So those are the empirical results, and I'll just say at that point we concluded that the tensile, the in-plane extension of the cell wall was controlled by the cellulose microfibril network. The indentation responses were controlled by pectin and cellulose networks. That plasticity was distinctive from creep, from this chemorheological creep, and we also concluded that it was not a safe approach to extrapolate from one type of assay to another assay. They reported on different kinds of properties of the cell wall. Cell wall has complex values. Now, these empirical results were interesting and defied a lot of predictions of these conventional models, but were unsatisfactory in that we didn't have a good theoretical way of understanding them. And so we started searching for a way of approaching that, and we collaborated with mechanical engineers to develop a coarse grain model of the primary cell wall. And so for this model, so I'll spend the rest of the time just talking about that model. And so for this model, we are asking what kinds of interactions between polysaccharides can result in the mechanical responses similar to the real cell wall. And what you see at the right here is the graphic image of the coarse grain model. One of those images is an early version of it, as visualized after the wall is equilibrated. So let me spend a minute or two explaining the nature of this coarse grain model. There's lots of different kinds of models of plant cell walls, and they have different features and different values. This one I thought was particularly useful. This approach I think is particularly useful for gaining insights and understanding some of the mechanical properties of the cell wall at a molecular level. So we begin by modeling the three major classes of the polysaccharides of the cell wall, that is cellulose, xyloglucan, and pectin. We model them as bead and spring models of polymers. So this follows a chain of beads linked together by springs. The beads in our model represent a 3 nanometer segment of the polymer. So that's about 6 glucose units long. And of course, it's a bit different for each of the 3 classes of polymers, but that's how this is represented. The springs are represent the axial stiffness and the bending stiffness. So there's 2 sorts of equations that characterize the bending and stretching properties of these polymers, and each of the polymers has its own set of stiffnesses of various sorts. And then the other important property is that we also model non-covalent interactions between polymers. So we know cellulose, for example, can bind to other cellulose polymers, other cellulose chains, and that's a strong interaction. Xyloglucans can bind to cellulose, so that's a strong interaction. Pectins have much weaker interactions. So those are modeled by Morse potentials, in which energy between 2 beads is dependent upon the distance between the beads. All right, so that's the model. It's very simple and it's very polymer based, and it's all non-covalent interactions between the components of the cell wall, the major classes of components. So we arrange these polymers in a simulation box in a way that's consistent with what we believe how the cell wall deposits these components. Then we equilibrate the components, and then we can do mechanical experiments on it. So first, the equilibration. So the initial setup, this image at label B here just shows the cellulose microfibrils as we initially put them down, and then after equilibration, they will spontaneously rearrange themselves into a network in which cellulose microfibrils bundle with one another and form a cohesive network. And the network has some of the characteristics, morphological characteristics of the actual cell wall, as visualized by AFM imaging. In here in color is the full model. So up on the right here in C, I'm just showing the cellulose microfibrils for clarity to show the network. The full model is just a single lamella now. The full model, but a single lamella, is shown here, in which cellulose microfibrils are shown in purple. You can see they form a reticulated network. The xyloglucans are shown in green, and you can see they assume lots of different conformations, many of which were seen in previous work from our group and consistent with other studies of xyloglucan. And the pectins are shown here in yellow, and they form a dynamic network which interacts with and binds to all the components, but those are fairly weak interactions. So the coarse grain model captured many aspects of cell wall structure and cell wall assembly, including cellulose bundling, cellulose network formation, and the conformation and location of xyloglucan and pectins. And so this was satisfying because it was, remember, just based upon the physical properties of polymers, very polymer-based model, and the likely patterns of deposition. So we're not fixing things to get a result. We put in the parameters based upon values from the literature and set this up, and presto, we get a model that resembles, with many respects, the actual cell wall. Now, to do mechanics, we set up a polylamellate system, as we knew the onion wall was polylamellate, and we use 4 lamellae with cellulose microfibrils initially oriented in different directions. You can see that on the right lowermost panels, 4 of those lamellae. And in D, you can see a top view of these lamellae. It's of course rather confusing because there's so many components there. And in F and G, you can see close-ups of these. So G is sort of a type of a cross-section at a slight angle. So you get a better sense of how these polymers arrange themselves in this polylamellate construction. All right, so once that's set up, then we can do some mechanical experiments where we stretch the wall and measure the forces and therefore the stresses that develop in the polymers of the cell wall, and get stress strain curves. And so I'll show you a movie of a single lamella. We did this with the multilamellar cell wall, the four lamella cell wall, but it's visually too complicated to see what's going on. So single lamella illustrates what's going on. And what I want you to notice is that there's dynamic stretching of the polymers, there's change rearrangements, there's changes in bundling. So this is a very dynamic model which carries out the kinds of changes we might anticipate the polymers undergoing when you're stretching a cell wall. So I'll show this movie once. It goes by fairly quickly. Sorry about that. And then I will show some stills just to emphasize some of the aspects of it. So here's the movie. Show it a second time. I lied. I didn't show more than once. Okay. And we could observe a lot of this many times somewhere. So in our science article, there's a movie like this in the supplemental material. So what you see is rearrangements of the cellulose microfibrils. You see dynamic stretching of xyloglucans. They let things go. And as you can follow by looking at some of these highlighted regions before and after. All right, so in the coarse grain model, we were particularly struck by certain kinds of changes in the cellulose microfibrils, and they're detailed in the paper. But we were attracted to those because a few years before we did the actual experimental observations of cellulose microfibril deformations during stretching experiments. We observed those by AFM, and that work was published. So again, empirical results, that work was published in 2017 in Nature Plants. And so the kinds of behaviors that we saw in this experimentally, well, it was also paralleled and matched by what we saw in the coarse grain model. And there's two of these behaviors that we think are particularly important for the mechanics. So one is the straightening of the cellulose microfibrils. As you stretch the cell wall, you can see a straightening response. And there's also sliding of cellulose microfibrils. So there's the slippage once the stresses in the cell wall build up. There's also other changes that are less important, we think, for the mechanics of the cell wall. So we could carry out the same sort of stretching experiment with our onion cell wall. In this case, we're carrying out a cyclic stretching. So we stretch the cell wall in 2 cycles, as you can see in that movie that I just showed. And we can get stress strain curves, both from the model and from the experimental results. The model results is shown here in blue. So this is stress versus strain versus some empirical results with 3 different data sets. And on the right-hand curve, you can see the slope of the line, so the modulus as a function of strain. And so you see the modulus is not constant. In fact, it gets steeper and steeper. So there's a stiffening response as the wall stretches, which, as you'll see, we attribute to the straightening of the cellulose microfibrils. And then some softening or some yielding kicks in, which is a consequence of the sliding of the microfibrils once a yield threshold is reached. The cyclic loading gives us some more data. So here's the experimental cyclic loading curves, and you can see the first curve in blue and then the second curve in red. There's an irreversible deformation. So there's a plastic deformation that occurs. And then the red curve, the second loading, if you repeat it over and over again, you see something very similar. So that's essentially an elastic curve, but there's the hysteresis between the loading and unloading curves. The unloading curves are shown here in the dotted lines. You can see that the wall is also, even after plastic deformation, is very stiff. These sorts of curves have been noted for a long time, but understanding the molecular basis for them has not really been approached in the past. The computational results give something very similar. Numerically, there are some differences, but qualitatively, this model has the same sort of thing. So there's a plastic deformation, and there's an elastic hysteresis that occurs in the second cycle. So the coarse grain model captures the nonlinear mechanics of the onion cell wall, including stiffening, yielding, hysteresis, and plasticity. Now, just as an aside, we also, when we did these stretching experiments, we tried varying the strain rate. And so that's shown. We do a pre-stress, so that gets rid of the plastic deformation. And then we repeatedly do the second stretching, and then it's a third and a fourth. And we change the strain rate by a factor of 100. And the curves are the same, right? Which is a surprise, because we always talk about the walls being a viscoelastic material, and viscoelastic materials, at least in my my thinking, should show a strain rate dependence if there's a viscous component in it. And yet these walls do not seem to show a classical viscous component. So just to double check whether our intuitions were working right, we took a pectin strip. These are fruit strips from the store, all made entirely of pectin, and did the same sort of cyclic experiments. And there we see, in fact, a big effect of the strain rate. So those pectin strips do, in fact, show viscous type of behaviors for viscoelastic materials. So the pectin strips show what we thought of as a classical viscoelastic stretching behavior, but the onion did not. And we might call this elastoplastic, but not viscous. All right, so back to the coarse grain model. One of the nice things about the model is you can interrogate the model and ask questions and get answers that are difficult to do experimentally. So, for example, you can ask, how is the stress distributed by polymers? And so I show here the stress distributed by xyloglucan, cellulose, and pectins, and you can see from this curve that it's almost all the stress is borne by the cellulose microfibrils. The other thing we can do is we can ask in this polylamellate cell wall, how is the stress borne by the different lamellae? And, you know, I tried to muddle through intuitively my thinking of this, and this provided me with a quantitative and very satisfying result, which is that the stress is dependent upon the orientation of the cellulose microfibrils, which intuitively you might think, but this provides a quantitative assessment of that. So the minus 15 degree lamella is the cellulose is oriented minus 15 degrees. So that's minus 15 degrees off of the direction of stretching. And so that is the stiffest component of the cell wall. The one which is close to right angle, so the 75 degree lamellae bears very little stress. And the intermediate, the lamellae with intermediate cellulose orientations has an intermediate stretching. So when you stretch this polylamellate cell wall, the stresses borne by that wall, at least according to this very simple cell wall model, the stresses borne by that cell wall are not evenly distributed on the lamellae, but very quite dramatically between the lamellae, depending upon cellulose orientation. You can see that result, the graphical result here in a different way by using we used a color map to map the stresses in the cellulose microfibrils and that their distribution, and with the 4 lamellae. And you can see here this lamella that's oriented closest to the direction of stress stretching has the hottest colors. So it has the largest stretching or the largest forces. And so the stress is not homogeneous. And it can be quite large. In the model, some of the microfibrils pick up over 800 megapascals of stretching of stress and that is 2 orders of magnitude larger than the computed stress of the whole cell wall. And of course, the difference there is stress is a continuum concept. The cell wall at this scale is not a continuum. The stresses are unevenly distributed. They're picked up larger by this fiber structure, which shows a very heterogeneous distribution of stresses. I found this aspect of this coarse grain model also intuitively satisfying, and could be quite important when we start to think about concepts of plant cells measuring the stress of the cell wall. So in the plant morphogenesis field, there's a large discussion about the stress in the cell wall serving as a guiding influence on the orientation of microtubules in the cell. And so how does a cell measure the direction of stress? And knowing the stress distribution in this sort of detail is important for those kinds of considerations. All right, and winding down now, this is the last slide in which I superimpose the stress versus strain curves for different lamellae with different cellulose orientations. And on top of that, I put in the major modes of cellulose motions. And so let's just take the blue curve. So this is one in which the cellulose is oriented in the direction of stretching. You can see a strong nonlinear elastic component, and there's a stiffening reaction, and that's largely where the microfibrils are undergoing straightening. And then at some point with a yield threshold of empirically the value as shown here, sliding begins, and sliding gets larger and larger and larger, leading to a fall off in the slope of that line. And so that sliding is resulting in plasticity and a type of yielding. All right, so winding down now. I think these traditional models of the primary cell wall, I think, are inconsistent with empirical mechanical behaviors, and also inconsistent with the mechanical responses of those walls to enzyme digestions. In contrast, the coarse grain model shows behavior that looks to be consistent with those sorts of behaviors, and it captures both the nanoscale behaviors of microfibrils, which in part have been confirmed empirically with AFM imaging, for example, and also the macro scale behavior, the stress strain behaviors of the cell wall. All right, so what's different about this model? This is a second conclusion slide. So in this model, the tensile forces are transmitted directly through the cellulose network, not via the matrix as an intermediary. The cellulose contacts, cellulose sliding, and cellulose straightening are key factors in the tensile mechanics, according to the model now. And xyloglucan and pectin backbones bear very little tensile stress. And I'll just add the one caveat is there is empirical evidence of xyloglucan being important for the mechanics in certain domains where there's a mixture between and tight binding between xyloglucan and cellulose. So there may be different kinds of junctions in the cell wall, some mediated by xyloglucan, some not. Okay, so there are, of course, lots of caveats to this model, and I won't detail them all. I'll just say that we modeled it after the onion epidermal cell wall, and the model gave us a lot of insights that were intellectually satisfying and that are testable. This model is one example of a primary cell wall. There may be other kinds of primary cell walls where things would have to be adjusted and tested, so how applicable it is. I would like to give acknowledgments to the great group of students and postdocs and collaborators who contributed towards this effort. They are listed here, and also support from the Department of Energy through 2 funding groups. And with that, I thank you for your attention, and would be pleased to answer any questions you might have.